And good evening, everyone. Welcome to Trolling with Logic. It's the 18th of January 2014. I'm still getting used to 2014. And it's 7 p.m. here in the UK, 2 p.m. Eastern Time over in the States. Just to let you know, you can call in later on in the show at the Skype contact Trolling with Logic. And as a reminder, when you call in, you grant us permission to use your likeness because the show is later uploaded to iTunes and YouTube for later viewing. So no funny stuff with the DMCAs. Also, the recording of this show is public domain, so feel free to copy, share and distribute it as you want. Just link back to us as a little courtesy. And I think that's all. That's the legal stuff. So quickly around the team and welcoming back after another long layoff. Wolfie, how you doing?
I'm doing good. Oh, it's great to be back. Good to see everybody.
All right. And Marty, how you doing?
Yeah, I'm fine, but my webcam isn't, so I apologize for that.
And as always, Sheila, how's you?
Hey, glad to be back.
And last but certainly no least, Kitch, what's up?
I'm grand. How's everybody?
All right. Yep, we're doing great. So you did have an intro for our guest role written up there, so take it away.
Yes, I've just written up a brief intro. I just want to welcome for her first podcast as Dr. Abbie Smith. She has defended her thesis on novel strategies to understand HIV treatment. She runs her own science blog on Science Blogs on ERVs, and she has debated for since a few creationists such as Charles Jackson and Steve Kern. Yep, that's just the brief introduction I just written up there.
All right. So as always, I think Sheila, you can take it away with the introductories. And sorry, let Abbie say hello to everyone first. Sorry about that.
Oh, hi guys. I'm really glad you all invited me to talk. And like he said, I just got my PhD. So this is the very first podcast that I am Dr. Abigail Smith. So,
The doctor is in the house. So Sheila, back to you. So we'll let you start with your introductories.
Awesome. Well, let me start with congratulations on your doctorate. That's awesome.
Thank you.
Can you give us a little bit of background? How did you get into the field that you're in? What made you decide to go the direction you did?
That's interesting. So when I was an undergrad, I was like a hardcore pre-med student. I was planning on going to medical school. I came from a small farming town, so I thought if you were good at science, you either became a physician or you became an engineer. I didn't really know any PhDs in biology. I thought they did like forest work and stuff. That's what I thought a PhD in biology did. But when I got to college, I was around actual PhDs in biology who did different kinds of biomedical research. And I got an opportunity to do a summer research program in virology and HIV research. And I only did it because I thought that having, HIV research on your medical school application would look really, really good. And then it turns out I loved it. I had so much fun. And I had more fun. It was more entertaining to do that than the stuff that I was doing in hospitals shadowing physicians. And so I was like, maybe I shouldn't be a physician. Maybe I should explore this other opportunity. So I took a couple of years off to be a technician, a research technician in an HIV lab to really see if, if I really did love this. Did I want to do this 9 to 5 plus every single day? And I totally love it. I don't want to do anything else. This is dream job, so much fun. How I got into HIV research.
Good story.
Yeah, no doubt. Where are you hoping that this field will take you? I mean, what is your? Is there like a specific? Are you? Are you working on trying to? Are you wanting to be part of like the team that finds the cure for HIV? Or I mean, what is your goal as far as work?
Really, as a scientist, I just love studying HIV as a virus. I think it's fascinating. It has evolved millions and millions, if not billions of years to be absolutely exquisite to do what it does. And just the way HIV does things is so different than the way, for instance, giraffes or oak trees do things. Just studying the virus itself, I think, is incredibly fascinating. At the same time, it's incredibly destructive. You don't read about oak trees killing 27 million people a year. So really, that is what all of my research focuses around, is studying the virus and hoping that when we understand the virus better, we can figure out how to make an effective anti-HIV vaccine. Because right now we don't have it. I've said this before on other podcasts. Like when HIV was first discovered, scientists were like, give us a few months, but this is a virus. We know how to make vaccines. We're on top of this. Just give us a minute. And here we are 30 years later without an HIV vaccine. And that is because HIV behaves in a manner different than the other viruses that we do have vaccines for. So just the way it replicates, the way it evades our immune system is different than, for instance, polio or chickenpox or other vaccines that we have made.
Right. I guess, can you explain a little bit the difference between, like, there's like a cocktail of drugs that people that are infected take. Mm-hmm. Can you kind of explain the difference between, is that more of a just kind of keep it under control sort of thing? What would be the difference between that and a vaccine?
Yeah. So the antiretroviral drugs are keeping the virus from replicating a lot, from making lots and lots and lots of baby viruses. But just the way HIV works, it's always going to be there. You can't get rid of every last virus, but the drugs control it. Whereas an anti-HIV vaccine, you would get before you were ever infected, and it would prevent the virus from ever getting a foothold, so it can't be eradicated. That being said, an idea that's been floating around is something called a therapeutic vaccine. So let's say you were already infected with HIV. We might still be able to give you an HIV vaccine that has a therapeutic effect. So if, for instance, we could figure out what the more aggressive variants of HIV looked like, even if you were infected with HIV, we could give you that vaccine, and then that would prevent the more aggressive ones from evolving. So we have the therapeutic drugs, a theoretical preventative HIV vaccine, and then a theoretical therapeutic HIV vaccine. Really, right now with the drugs, that's our best option. The anti-HIV drugs, when people take them, it's great at extending lifespans. People who are infected with HIV, their lifespans are approaching those of uninfected individuals because the drugs we have now just work really well. And actually the lifespans of individuals in a community where the people with HIV have access to the drugs, the overall lifespan is increasing because other individuals' chances of acquiring HIV are reduced when everyone is on the drugs. So really right now, the goal is to get as many people on the drugs as soon as possible to help control the epidemic.
Right. Is there like an issue with the patients who are on the drugs? Does the virus kind of adapt to that, to where the drugs become as not quite as effective or eventually ineffective against it?
Sometimes. So you know how I was saying that HIV behaves a little different than the other viruses we've dealt with? Well, what happens is every time HIV replicates, each one of its little baby viruses is a little bit different than the parent strain. And so when that goes on over and over and over, what you get is a big cloud of viruses where they're all related to one another, but each individual one is distinct. They're all individuals. So what happens is you will give someone an anti-HIV drug, let's say AZT, and that will wipe out most of the cloud of genetic variants. but it's not going to get all of them. Just by chance alone, there are going to be variants in that cloud that are resistant to AZT. And so what we found out very soon after AZT was developed was that you can't give someone just one drug at a time, because then all that happens is you wipe out all of the viruses that are unable to deal with the drug. You're left with a population that's resistant, and then the population expands to form a resistant cloud of viruses. And so that's the idea behind having people on 2, 3 more drugs at the same time, is that it reduces the chances that there's going to be a variant in there that can resist all of the drugs you're throwing at it at the same time. But ultimately, there are variants in there that can. So really, it's a lot of luck how long a patient can take the drugs before the virus becomes resistant. Because in one person, it could be a matter of days. Another person, they could go their entire lives and not develop the resistance. But it's a numbers game.
Okay.
You mentioned the therapeutic vaccine. Are there any possible targets for vaccinations that would actually be... Sorry. Are there any targets on the HIV cell or the HIV particle that would be an indicator for aggressiveness?
Yes, actually. So you have the HIV virus, and there's the little protein sticking outside of the virus, and those are the proteins that stick to the host cell to fuse to infect the new host cell. That's called the envelope protein. And what we found is that the fitness of the virus follows the ERV. So if you have a really aggressive virus and a more benign virus, and you switch their envelopes genetically, you can make the benign guy more aggressive. So that's actually good luck for us, because that protein on the outside is the only one that can be a target of antibodies. So it's what our antibodies will stick to on the virus to keep it from infecting another cell. So that's good news. And the bad news is that particular protein changes a lot. Like, it can tolerate extremely high levels of mutation and still maintain functionality. And that's actually what one of my papers was on, was that you could really radically change that protein and it still operates just fine. And that's really, unfortunately, logical and what we see in nature, and why it's so difficult to make an anti-HIV vaccine.
There was a question from the chat room from one of our watchers, I guess. How about the bone marrow transplant? There was a guy in Germany, if I'm not correct, that was cured of HIV.
Totally. He totally was. So what happened was some people with HIV get cancer. I mean, just people in general get cancer. And this individual had a blood cancer of some kind, so he needed radiation, chemotherapy, and a bone marrow transplant to get rid of his cancer. And it's really what physicians do, is they treat kind of a hierarchy of what's killing you right now. And the cancer was killing him more than the HIV was, so they treated his cancer. And it just so happened that his match for a bone marrow transplant had a weird genetic quirk. This donor lacked one of the co-receptors that HIV needs to infect new cells. It's not the only one HIV can use, but it's one of the dominant ones. It's called CCR5. And just naturally, some people have this mutation. They don't have this protein, and they live totally normal lives. It's kind of dispensable. So this physician gave this HIV patient basically new immune cells that HIV would have a really hard time infecting. And the end result is this individual, basically, they can't find HIV in his system anymore. So the radiation and chemo killed lots and lots of the HIV-infected cells, even the ones that weren't actively producing virus. And then with the bone marrow transplant, replaced it with fresh blood cells that couldn't be infected. So it's fantastic. This is great. Why aren't we doing this with everyone? Well, because I think it's something like 50% of the people who get this transplant on the table. So if someone isn't in danger of dying of HIV, no physician is going to be giving otherwise healthy person a procedure that could kill them. So what some people tried here in the U.S. was it's really hard to find a bone marrow transplant match, immunological match in the first place. So trying to find one that has this genetic quirk at the same time is even more complicated. So they tried something a little different. So normally when someone with HIV has one of these cancers, they take them off those antiretroviral drugs because they're really aggressive. They're hard on your liver, hard on your kidneys, hard on your system. So they take them off the drugs, treat the cancer. Once they're feeling better, they put them back on the drugs, but they're still HIV positive. What these guys did was when these individuals were going through their cancer treatment, they kept them on the antiretrovirals the whole time. And then ultimately, at the end of the study, they had 2 guys who had no detectable HIV. They were still on the drugs, the drugs that repress the replication of the virus, but no virus, which is great. But then their physician did something incredibly stupid, which was to take them off the antiretrovirals. That was really dumb. And in all fairness, this guy is a physician. He is not a PhD. So maybe he just genuinely didn't understand the implications of taking these patients off the drugs. The virus, of course, flourished in the absence of the antiretrovirals, and rebuilding that cloud I was talking about. So instead of having everything suppressed, the cloud rebuild, and now you try to get them on the drugs again, and it's really complicated. These 2 guys could have had probably an extremely long life, long suppressed HIV, and now it's been allowed to bloom again because their physician took them off the drugs. So that was really, really disappointing. But as to why it's not ultimately going to be more widespread, well, it's just a really complicated procedure. It's a really dangerous procedure. We can't go to a village in Zambia, a farming village in Zambia, and give a whole bunch of kids bone marrow transplants. That's not going to be a long-term solution to the HIV epidemic.
The same guy had a second question. Are there signs of evolution making people resistant to HIV?
Here's the problem. If HIV had crossed over into the human population, been able to spread around the world the way it had, let's say, 5,000, 10,000 years ago, then yes, I think we ultimately would have seen evidence of that. I was saying that some people don't have that receptor. They don't have a functional version of that CCR5. That probably would be a lot more prevalent today than it is. But what happened was HIV crossed over, I guess you term it maybe a post-Darwinistic medicine era. We don't allow nature to just take its course, which would be necessary for the humans that were most susceptible to HIV to die. And the virus that doesn't affect us as much go untreated. We don't do that. Like, we have to treat patients. We're not just going to let people die and say, oh, well, in 500 years, humans will be more resistant to the virus. See ya. That being said, we can actually kind of see it in the virus. So it's something I wrote about recently. People are always asking me, is HIV evolving to be more deadly, more aggressive? And ultimately, it's not. When we look at the analog in non-human primates, SIV, we don't see SIV that kills monkeys in 2 weeks. What we see is a virus that is maintained in the non-human primate at pretty high levels and is passed to other primates through sex, to baby monkeys during monkey birth, childbirth. I don't know what you call that. But when you think about it, it's like, what good is a dead host to a virus, and particularly this kind of virus that doesn't live outside of the host very well? What the virus wants, if I'm allowed to personify them, is to have a host that is otherwise healthy, out there having sex, having offspring, and being passed to the new host. They go on to pass it to new host, a new host, a new host, without killing them. So really what we kind of see in the virus is that admittedly limited numbers of viruses have been tested, but the newer guys aren't as aggressive as even the viruses that were infecting us 20 years ago. So we can see that.
Who's next with the question?
Sure, I can ask a question. Just to change topic a little bit. You've debated some creationists, such as I said in the beginning, Charles Jackson and Steve Kern. How did you get into doing these type of debates, and what's your general experience of them?
My very first one was actually with an HIV denier, and I just end up having friends who somehow have these connections, and they'll say, hey, like they're friends with churchy people, or they have some friends who are a little wooey, not so scientific circles, and they'll have a friend just gushing about this HIV denier, this anti-vaxxer, this creationist that was coming to town, and my friend would be like, hey, I've got a friend who believes something different. Let's set up a debate and have some fun for everyone. And so that's how I ended up getting into that, was just I was around when people got the idea. I think it is an excellent opportunity, especially for students, to really get a different, what's the word I want to say? It was a different kind of knowledge that you have to just know something and to be able to present it to a lay audience, and to really assimilate lots of information from different classes, from different textbooks, from different parts of your life even, and assemble them together in a presentation like that, in a debate. I think it made me a better graduate student, honestly, in that I was looking at things outside of like directly what I needed to know for my dissertation. I was still keeping my eyes open on the field of endogenous retroviruses and evolution, even though, specifically I was studying HIV. I had a broader view on my very narrow topic. And also, it was just a great outreach opportunity. I see debates as a chance to educate the public about science, and it's obviously something they're interested in, whether it's pro or con at the time. Give them a chance to learn something. And then pretty much every debate I've been in, I've had someone from the other side come up and be like, you aren't at all how I pictured the people on the other side. So the debate with Charles Jackson, there were these kids in this church who had been told their entire lives that scientists and evolutionists, whatever, were these evil, I don't know what kind of caricature they were taught over the years, but they came up to me and were like, but you're so nice. Like they couldn't believe it. And that made it all worth it. Like that debate with Jackson was so long and so frustrating. But the second those kids came up and were like, but you're so nice, that was all worth it. Because, just putting that little spark, that little idea of maybe what these people have been telling me isn't all that accurate. Getting them ideas to go out and investigate things on their own totally worth it.
Can just butt in there. I wanted to ask your quick opinion. Have you heard of a guy called Mike Adams?
Yeah, yeah. Is he like one of the?
Yeah, he's one of the main kind of. I was just wondering what your him and his Natural News website and that, if you'd come across it and what your opinion was.
I actually think I came across some of his articles on a different website. Right. I was Googling news on endogenous retroviruses just to see what kind of press releases universities had released on some new pop research. And I found this website that was absolutely positively insane. Like, I've been doing this a very long time, and this was one of the weirdest, most hysterical websites I'd ever seen. There was one article was like on what colors you should wear to protect yourself from assault from psychics. And they literally called they were like psychic condoms is what they called, these colors that you wear. I'd never seen that before, but I know they had articles there from Natural News and those kinds of websites. So,
that sounds like something from Spirit Science.
That sounds like something. That sounds similar to like the Mormon magic underwear.
This was crazier, dude.
Oh, they just keep getting crazier. Yeah. They always do.
Who's got the next question there? Sorry,
I guess I'll say something.
There you go.
Dr. Abbie, I'm just letting you know I'm, of all the tutorials, I'm not the scientist. I'm the historian at best. But amazingly, 10 minutes before you ever mentioned CCR5, I was literally reading about it for probably the first time. I was like, holy crap, she just brought it up. And so I'm reading about its possible origins and the mutation, why there's a very small portion of Europeans that carry it, only about 2% in Central Asia, and it's practically almost absent in the East Asias, Africas, and tribal Americas, and it's probably due because of the Black Plague that caused the mutation. But I was also reading on sciencefrontiers.com, if one inherits this gene from both parents, they will especially be resistant, if not immune, to AIDS. Has that ever come across your table?
So the connection with the plague, I think, was something they originally might have seen, but then once they went back and investigated it further, it wasn't a connection. I'm trying to think of the specific details, but I think they ultimately dissolved that. But as far as being totally immune to it, no. So most kinds of HIV like to use CCR5 as the co-receptor to get into cells, but it's not the only one by any means. And there's another one, CXCR4, that it really likes to use. And that's actually a more aggressive kind of HIV. And it's what we see evolving in some end-stage AIDS patients. So no, don't know. If you get tested with 23andMe or one of those DNA testing sites and it says that you're homologous Delta CCR5, don't think you can't get infected with HIV, because you totally can. And if anything, it's a kind of scarier form. But your chances of becoming infected are less, yes, because most people have the CCR5. And so if you're exposed to that virus and it's got no cells to infect, then that's the end of that. But no, you're not totally immune. Don't think you're immune.
All right. My last question would be, do you have any scientists as role models or rock stars?
No. there are big names in my field, absolutely. And there are names that are incredible at what they do in HIV research. They have a thing, they do their thing, they're great at their thing. But I don't know whether we necessarily do the rock star thing so much, actually in science, because I don't know. I don't know because. I don't know, because
It's probably not the best term I could have used, but it's something that I say from time to time.
Yeah. Well, there are some really nice, really intelligent, really innovative people in HIV vaccine world. And I mean, it's just like anything. There's the cliche, arrogant, full of themselves researchers. There's the really humble ones. But a rock star, I don't think we have the personalities to carry the rock star label. I think even the extroverts among us are introverts in normal populations.
Okay, we do have a caller. Henry, are you there?
Yeah, I'm here.
All right, yeah, you're on with Abbie, or Dr. Abbie as we keep calling.
Yes.
Yes, Abbie.
You just have to do it right now. She just got her PhD, guys. Let's just call her doctor.
One step closer to world domination.
Yes.
So what's your question for Dr. Abbie?
Well, it might be a little off topic, but it's in relation to retroviruses and evolution. I wanted to know if you were ever thinking about creating a book. I know you've been busy lately, but if you wanted to create a book on that subject.
Here's the deal. I often get emails from people wanting a good layman's introduction to viruses. And I don't have a good book to offer them that give you really the basics of the field. And I think one of the reasons of that is that, if you pick two viruses at random, hepatitis C and influenza, there are so many things different between those two viruses. They're both viruses, but they're radically different creatures. And so to fit the basics in a small, readily consumable book is definitely difficult. But it's something that people want at the same time, and it's not out there. So, maybe at some point, I don't know if I'm the best person to do it, but I think it should be done. And there are cool books out there. So Carl Zimmer's Planet of Viruses is a great little book that contains lots of different articles that Carl has written that are nice little stories. You don't necessarily learn the basic fundamentals about a virus, but you read stories about them and things the viruses have done and just interesting CDC findings and things like that. So if you want just a little introductory book on viruses, that's the one I would recommend, is Carl Zimmer's Planet of Viruses.
Anything else, Henry?
I just wanted to thank you for that, and I think you would do good on a book like that. I know you've been busy, writing your thesis and everything, but I think you would do good on it. I watched some of your videos, and I'm usually kind of a freak, I would say, when it comes to like viruses and keep my hands clean and all that stuff. And I watched some of your videos on YouTube in connection to the Thinking Atheist, and I thought, it was like a revelation, so to speak. And
we're doing cool stuff with viruses. Like they aren't as scary as a lot of people make them out to be. Like they're really, the way we look at them now is more of tools to do our bidding as we go to the world domination thing again. Yeah. So I think maybe there is an audience for that, to educate and demystify viruses for the public. Yeah. It's a good idea.
All right, Henry.
Yep.
Is that everything? Yeah.
Yep. Thank you very much.
Thank you very much. Thanks for your call.
Thank you for calling in.
Okay. A few chat room questions. Mortimer, you've been very silent, so...
Yeah, that's because this has to do with biology, and I don't know a damn thing about biology.
He's a physicist.
I'm a physics geek myself, so... Okay.
Gotcha. Well, I think there's not... like the quasi-species population model for viruses, there's a lot of math in that. And there's a lot of randomness and chance that throw everything off when we're doing experiments. So I think that there's a little place for physics. you can inch your way there.
Yeah, well, there's room for physics in virtually all fields of science, but there's more or less of it. And, plus, I just don't find biology very interesting. Sorry. I'm glad that
that's
there are
okay.
people who
I
do
didn't
think it's
know.
interesting, but I don't.
When I was
but
in
anyway,
high school, I hated it. So, like, I thought it was really boring.
Yeah.
Biology in high school. Like, I thought that was like the more easier classes that you would take to get your A, and I would know I was taking the physics and advanced this, that, or the other. It was totally with you. It was really just me falling in love with viruses that got me into biology the most.
Yeah,
I'm with you on it, man.
I have a similar story to go off on a bit of a tangent when it comes to didactics of physics, because I'm a teacher and I had no idea how fun it would be to write a thesis physics didactics. So I wrote about the use of analogies, and I'm actually continuing with that and getting a PhD. I just haven't had the opportunity yet, but it's something I'm considering. But so it's kind of similar to your story, I guess. But anyway, there was a question from the chat room. In how far? I think there's an English fail here. This idea of HIV being a gay disease.
Oh,
is it more is it disproportionately common among gay people, or is that just a myth?
It is true, but it happened in the sense really more of a founder effect. If you know what that means in evolutionary biology, it's just sort of a side effect of where things started. And it's a side effect of where things started in the Western world. So in Africa, it is absolutely a heterosexual disease. Actually, it's married women who are I don't know whether it's most at risk, but highly at risk of acquiring HIV due to infidelity of their partner, going off and having unprotected sex with prostitutes and then coming home and infecting mom not knowing, mom having babies are infected because she wasn't on anti-retrovirals. Mom finds out she's infected when she is having baby, and then husband comes home and says, oh, you're infected with HIV, you cheated on me, and now we're getting a divorce, and mom is infected with HIV by herself with kids. That's the situation in Africa. It has absolutely nothing to do with gay anything there. But just where the virus was first introduced in the Western world, that's why we call it a gay disease here in the U.S. and I guess in Europe as well. But yeah, if that initial introduction had been via heterosexual contact, then technically being homosexual would have been protective because you wouldn't have been exposed to the infected population. But actually, really, we should thank the gay population because they were the huge, unignorable red flag that told the rest of the world that something bad is happening. When all of these otherwise healthy males, like adult males, were coming into the hospital sick, if not dying, of these diseases that they should not have been sick or dying from, the secondary infections that you get with late-stage AIDS. That is what raised the call for scientists around the world to start looking for this virus. And once we found it, then we could start treating it. But no, it's not a gay disease. There is an increased risk of transmission via homosexual male contact. But you can still get it whether you're on top or bottom or gay or straight. Wear condoms. Everybody wear condoms. My job would be so much easier if everybody wore condoms. HIV does not go through condoms that are not broken. It can't, it's inert. It can't swim through the holes or anything. So HIV cannot go through a condom. It does not evolve resistance to condoms. Condoms are great. That's my takeaway message.
Nice.
Stay in school, drink milk, wear condoms.
That's good advice.
was
That's
just-
probably
Sorry.
better advice than you practically hear from like the Catholic Church.
Yeah.
Yeah.
Just
But
on-
I don't know
on
whether
that,
they're on board or off board with the whole condom thing. They got a new pope. The old pope would be like, no, never use condoms. And then the old pope, for at least one brief moment, said, well, it could save the life of the uninfected person in a relationship. So it was treasuring life to wear condoms in that particular instance with a monogamous couple where one was infected and one wasn't. And it took them that long. Like, HIV has infected the human population for a very long time. And I think it was maybe last year, the year before, we finally got the Pope to say something. But
It's just
Yeah, I
another-
don't know if it was the what was it? What was it, Ratzinger or one of those pubs? But it was one of the very recent ones. I think it was Ratzinger. He was the only time he spoke of condoms, but he often spread not just the message of don't use condoms. He spread lies that they
And
increase
misinformation.
the chances
Who knows?
increases the chance of getting AIDS, but that is pretty much horseshit. Even if you just have access to look up and verify that information, but that scared a lot of people in Africa, especially the very devout religious ones. It's like,
Yeah.
okay, no, can't go anywhere near a condom now.
It was just on that note, Abbie, what's some of the craziest kind of ideas of how HIV is spread that you've heard?
Well, when people hear that we got HIV from monkeys, immediately everyone thinks that people were having sex with monkeys because you get HIV from having sex with people who have HIV. Therefore, we got HIV from having sex with monkeys. Probably not, is the way I'm going to put that. A more logical hypothesis for how it crossed over into humans is that in Africa, they hunt monkeys for food the same way, in the U.S. we hunt deer and turkey, and I don't know what you all hunt in the wild in England and Scotland. Quail,
They go fox hunting.
Yes. You hunt it for food. It's food. And anyone who has hunted or fished knows that you cut yourself, taking the line out, butchering the animal. There could be an opportunity for exchange of blood in that sense. And really, this crossover between primates and humans probably happened a lot over the course of human history. The difference was when HIV crossed over this time to create the pandemic we see today, we had a way for someone to get on a ship, get on an airplane, and go from Cape Town, South Africa to Tokyo, Japan. Humans didn't do that 5,000 years ago. But in a worldwide population like we have now, it was easier for this random event to turn into a worldwide event. So that's probably what happened. And it happened at least 3 times because we have 3 different kinds of HIV-1. And then it happened another kind. So that was chimpanzees. So 3 times somebody killed a chimpanzee and got infected with SIV. And then there's a different kind of primate we got HIV-2 from. And it's not as aggressive because it was a bigger leap between the 2 hosts genetically. So it didn't adapt to humans as well. The ideas of where HIV came from that are also totally crackpot. So HIV deniers, some HIV deniers think that HIV does not exist at all. And some HIV deniers say that HIV exists, but it doesn't cause AIDS. And then there are some HIV deniers who say HIV exists, it causes AIDS, and it was like a government conspiracy. They did it on purpose. A retrovirus is a very poor choice in weapon. Like, you get infected and, hey, you might die in 10 years, or you might get hit by a bus before that, or you might live your whole life. without anything negative happening at all. Boo. I mean, it's just, as far as weapons go, and especially weapons, our government has at its disposal, I think a retrovirus would be a very stupid choice. And also, like, no one is naturally immune to HIV. So, like, smallpox was a good weapon because the European population was already, fairly resistant to that. So you come into a totally naive population, you can wipe them out. Everybody is susceptible to HIV. So I'm going to attack you and I might die too in 10 years. So it's stupid. We didn't even have the technology to do that. Like, evolutionarily, we know when HIV crossed over into humans. It was about the turn from 1800s to 1900s, somewhere around there is when it happened. And you're telling me that they had the technology back then to genetically modify retroviruses? Are you kidding me? Like, we don't have clean water, but we've got this kind of technology. No.
Well, yeah, but of course you're going to say that because you're part of the conspiracy, right?
It's true. It's true. And then there are some people who say that it's via vaccines. We were raising all of these monkeys to make vaccines or something. A polio vaccine is what some HIV deniers say. And we it wasn't on purpose. It was accidentally introduced HIV to the human population. But again, these are all, I will give them this, testable hypotheses. So the deal is, we know, like, through evolution, through genetic testing and phylogenetic trees, we know the exact colony of chimpanzees where we got HIV from. And those particular chimpanzees weren't ever used in any kind of vaccine production or whatever the heck these HIV deniers were saying. Yeah, that's actually a really cool story too. So chimpanzees don't swim. Like, they don't like to get in the, I guess, alligators and crocodiles and hippos and monkeys don't like to swim. And so in Africa, they've got, like, very distinct populations of chimpanzees that are separated by water. And so what the scientists did is they went out and they collected a whole bunch of monkey poop. And then they sequenced the viruses in the monkey poop and compared them to each other and compared it to human viruses. And we figured out which colony of chimpanzees had viruses in their poop that matched the human viruses. I thought that was pretty cool. I didn't think to do that with monkey poop.
And there goes everyone's conception of science being a classy job, just thrown right out the window.
Nothing glorious about it. I guess it just went out with Walmart bags and picking up monkey poop. So,
as they say, a quote from one of my favorite movies, Evolution: "Sometimes science stinks."
Yeah.
At least you can say you're not taking shit from anyone.
No, we're stealing shit from monkeys.
All right.
The precious poo.
All right, Marty, you can handle some more of the chat room questions as you're the quietest tonight.
Yeah. Okay. How common is HIV in the Western world?
I don't know. I guess we could look it up on the CDC website. It depends on a lot of where you live. It can be more rare or more frequent, I guess, depending on what city you live in. It's not, in the Western world, it's not as big of a deal as it is in Africa, as it is in Southeast Asia. So it's something that those of us in the U.S. and Europe can kind of forget about because, we don't, it's not something we face every single day, and it's not something, it's totally ravaged the African nation. I mean, it's killed a generation of people, left generations of orphans, and it's something other countries have to address in the way that we don't really have to. We have access to the drugs if we get infected. Yeah. We have access to testing if we suspect we've been exposed to it. And that's just really a luxury that still a lot of places in this world do not have. But the actual numbers, I don't know. Depends on where you live.
Yeah. Okay.
I'm
CDC will
willing to
definitely have the numbers.
I'm willing to bet money that there are more people per capita with HIV in areas where they, for the large part, teach abstinence-only programs.
It's been a huge problem in the South, and specifically African American populations in the South in the U.S. It's not something they talk about. Being homosexual is not something that's talked about, and that is not the way to address HIV. You have to have a very frank discussion of sexuality, prevention of HIV, and really, it's HIV is frequently, the top of the list of why people talk about this. There are other sexually transmitted diseases that are deadly. We're talking about hepatitis C, anything like just frank and honest discussion of sexuality and prevention of STDs and pregnancy and that sort of thing is what can help stop HIV. In the countries in Africa where they've had very aggressive education, pro-condom campaigns, they see a decrease in their HIV rates. That's fact at this point. So anytime they stop, the funding dries up for a brief moment. George W. Bush actually had a really good HIV funding platform set up, and recently they tried to change it so any institution in Africa that had lots of family planning and catered to prostitutes and that sort of thing, they had their funding yanked if they didn't teach like abstinence and that sort of thing in these clinics. And they ultimately got that reversed, so everyone got their funding. But just the idea that it is so frustrating as a researcher, and like I said, condoms, everybody, gay, straight, whatever.
Just condoms and education. It's like the same thing when they start spreading misinformation about vaccines. It's only when you see that in counties where they largely just refuse to vaccinate the kids, that's where they have outbreaks of measles and whooping cough, and it's happening again, I think, somewhere on the eastern seaboard, I'm guessing. So yes, education and condoms. That's a great way to go.
Education and prevention. Yeah.
And prevention, yes.
Okay, Marty.
I just have a
have
a quick question that I just want to ask. It's relating to HIV vaccination. What kind of vaccines are leading the field? Are most promising? Are they DNA vaccines, attenuated virus, or dead virus?
We have tried everything, is what I'm going to say. We've tried absolutely everything, and nothing has really worked out the way we had hoped. Sometimes things have actually turned out worse than what we hoped. We actually had some vaccine attempts where the people who got the therapeutic vaccine and not the placebo actually had an increased risk of contracting HIV than the people who just got the placebo vaccine. So what's the most promising right now? I would say really the question that needs to be answered at this point is, what do we put in the vaccine as the target? So like you said, there's lots of different ways to deliver the target. Killed viruses, genetically modified viruses, just the DNA to code for the virus. But what is that? What's the actual code of the DNA that you put in? What is the actual killed virus? Because with HIV, there are just so many variants. Why are you picking X instead of A, B, C, D, F, G, H, I, J, K? Why are you picking that one? And the question really is now, we've got lots of ways to generate anti-HIV antibodies and anti-HIV cellular immunity. What's the best target? Because what we have currently chosen as targets hasn't been effective. And so that's actually what part of my research project is looking at people who developed broadly neutralizing antibodies. So they're infected with HIV. There's nothing we can do at that point. But some people naturally make the kinds of antibodies that we want to make in a vaccine. And in a vaccine, we want antibodies that don't only neutralize this virus, but they neutralize all of these viruses, because you aren't going to be exposed to only one kind of virus out in nature. So I'm studying those individuals and trying to look at exactly what happened with the virus and exactly what happened to the cells that generated the antibodies, to see if we can mimic that through vaccines. Because right now we don't. You say what's the best one out there? We don't have a best one. We have lots and lots and lots of attempts that we've learned lots and lots and lots of stuff from, but nothing that we can take out to protect anybody today.
Okay, Marty.
Okay.
There's been loads. They're flooding in, so
Yeah,
hopefully
there's a bunch of questions, and I'm going to try to keep up. I definitely won't be able to keep up. Can HIV kill you directly, or is it always the secondary diseases that get you?
It's the secondary diseases that get you. So with retroviruses other than HIV, what they're known for.
Pause. Biology. What's a retrovirus?
Yeah,
a retrovirus. So like humans, our genetic is DNA.
Yeah.
Right, and so the DNA gets turned into these RNA messages, and these messages get turned into proteins that build up us. Retroviruses carry their genome as RNA, so that's like a message to us. That's not storage, but to them it's storage. And then once they infect a cell, they turn their genome into DNA and stick it into our DNA. So our cells just bop along thinking, oh, that's DNA, that's me. And that HIV has capitalized on that. And it's not just HIV. There's lots and lots and lots of different retroviruses. And a significant portion of your genome, your human genome, is actually retroviral. But that's a different story for another day. But there are lots and lots and lots of different retroviruses. And so what was the question again? I got lost.
Yeah, the question was, is it all, can HIV kill you? Or is
kill
it
you.
Just,
okay, so there are different kinds of retroviruses in different animals all around the world. It's not just a human-specific thing. It's not just a monkey-specific thing. It's lots of
because I keep hearing these things about ERVs. That's like parts of your genome that come from
endogenous retroviruses.
Yeah.
But there's lots of different kinds of retroviruses, and traditionally what retroviruses are known for is causing cancer, because you know how they carry their genome as RNA and it goes into DNA and it goes into us. You can't just like plop stuff in the middle of the human genome or the mouse genome or whatever at random and not cause trouble. And frequently what happens is that retroviral DNA plops into like a really important part of your genome, and that cell turns from a host cell into a tumor. So we've got like mouse memory tumor virus that it just inserts over and over and over as it's infecting cells, and eventually it causes a cancer. So those can kill you directly. HIV, though, and other lentiviruses, it's different flavor of retrovirus. They don't traditionally cause tumors. So it's not like if you're infected with HIV, you always get a certain kind of cancer. You might become more susceptible to other kinds of cancer because it's totally wrecked your immune system, or you're more susceptible to an infection. It's the secondary infections that really get you more than the virus directly. So it's not really good having the virus around either. People think that your immune system is impaired when you have HIV, and it is, but also it's like a hyperactivated state as well. It kind of exhausts your immune system when you're infected with HIV. But I don't know if anyone or any person is actually killed by HIV. That's a cool.
Okay.
Excellent, Marty.
Actually, I just have a question just leading off of that. What's the actual most common cause of death due to HIV infection? What's the most common secondary infection?
Oh, I don't know about that. I think, again, it depends on where you live and what you get exposed to and what kind of treatments you have. So someone in the U.S. that has access to really good medical care might not necessarily die of pneumonia, but they might. But that might be more prevalent in an area where they don't have very good medical care and they can't treat the secondary complications you get. Yeah, I don't necessarily know that, but I think, again, that's one of those questions that'd be different depending on where you live. So we can look it up. I think those are totally stats that the CDC and the World Health Organization would absolutely have if you want to investigate that.
And HIV infection, it affects CD positive T cells. These are those helper T cells that aid in the immune response. But is there any other cells that may be affected indirectly or?
Also, there are macrophages that they can be infected directly by HIV. But you have a lot, that's not as big of a deal because losing your CD4 T cells means it's really hard for you to make good antibodies because that's what that cell does. It helps the B cells make antibodies. And then if you want to start a fight in HIV research world, say that there's another kind of immune cell called dendritic cells. There's a fight over whether those cells are actually infected or whether they're just capturing the virus and then passing it off to other immune cells. And you will, that sounds like nothing. You will get a knockdown, drag out fight if you bring that up at some conferences. But yeah, it has, and like I said, having all of these viruses around and your immune system responding to these viruses, and then part of your immune system, getting exhausted and dying, it really does wreak havoc on your immune system outside of just specifically those CD4 positive T cells. So those are one of the prime direct targets, but it absolutely has widespread effect on your whole immune system.
Yes, I actually read a paper on that just from when I was doing my immunology in my final year in my undergrad, about DC-mediated infection of CD4. I thought that was a very interesting paper. Is there actually why is that so contentious exactly?
Well, because replication. Some labs infect their dendritic cells. They say, look, they're infected, they're infected. Another lab takes their dendritic cells and said, these guys aren't infected. I don't know what you guys were doing, but these are not infected. And they're like, no, they're infected. Look, they're not infected. Ours aren't. It's not when we do it. And so we can't currently come to a consensus. Because it's hard to tell whether a virus is just sticking to a cell and hugging a cell or actually infecting it and producing more viruses. It's more complicated than you would think it is, but it is. It's a fight.
Okay, Marty, back to you with the chat room questions. I think we've got so many now. I think we'll try
cool. I've got a blog. If we don't have time for these questions now, when you guys get this uploaded, I'll put it on my site, and people can definitely ask questions whenever. Or if you guys have a place,
we do as
well. Put that in the comments too, and I will check that and try to answer questions there as well.
Oh, that's great. So
okay, this is a question that's actually interesting. The answer should be pretty obvious even to me, but the question of why, however. Can HIV be cured by filtering all of an infected person's blood? I'm going to go ahead and say no, but
it's not just in our blood. So cells can get infected, and then they aren't just constantly circulating in our blood. They'll go out and they'll hang out in our dendritic or in our digestive system because they have other jobs. They don't just constantly flow. They look for pathogens in something we've eaten that's bad, or they'll go up and just hang out in our brain and just sitting there waiting to be exposed to something that says, hey, something's wrong with your brain. We need the immune cells up here to kill this thing. And that's part of the problem too, is that we've got these cells infected with HIV. They aren't producing virus. These are called latent cells. And then they go into our tissues and hide. And And that's really why there was hope that those patients that went on radiation and chemo and all that, like, you could kill all of those virus or all of those infected cells that were hiding in tissues and not just in the bloodstream. But apparently they didn't. Yeah. No, not silly, but it's a very good question.
Yeah, there's a lot of. I mean, those immune cells, don't they come from the bone marrow or something like that? Or
yeah, that's where they start.
Yeah.
But they're everywhere, and so they get infected. They migrate somewhere else, and that's a problem for treatment too. Because just because you take a drug doesn't mean it's going to get to your brain at therapeutic concentrations. You know, it's got to cross the blood-brain barrier. It's got these infected cells migrating can cause a lot of trouble.
Okay. Just got another question from someone who says they're gay and they want to know if there's a rational basis for refusing homosexuals to donate blood.
Well, every person who donates blood, it's tested. It's not only tested for HIV, it's tested for hepatitis C, it's tested for cytomegalovirus. Blood that gets donated runs through an extensive level of testing because no donation body wants to be the ones that infected someone else with HIV. Hepatitis C. Actually, hepatitis C is the thing I'm more scared of than HIV. We have antiretrovirals for HIV. We don't really have good options for hepatitis C. So really, that's the one I'm more scared of. But rationale, no, it all gets tested. And testing is widespread enough just in like the general population. I think if a HIV negative homosexual knows their negative status, nothing wrong with it. I mean, I don't see a reason for them to necessarily be banned.
They do. They refuse blood donations from gay males. And
yeah.
I think that's why he was curious as to the rationale behind that, especially since they do test everything that they take in.
Maybe back in the day when people weren't getting tested and there wasn't so much awareness and there wasn't so much prevention, we know how to prevent transmission of HIV now, that it was a valid concern. But now not so much. And actually, we've gotten to the point too, related but not, where HIV positive individuals can donate organs after they die. So if you die in a motorcycle accident or something now, you can actually donate your organs to other HIV positive individuals. In my head as a scientist, I'm thinking that's not a very good idea because just because I'm infected with HIV and you're infected with HIV, they could be radically different kinds of HIV. Yours might be more prone to becoming resistant to the antiretrovirals. They can recombine and cause issues. If you're getting an organ transplant, you're going to be on immunosuppressive drugs, which it's complicated. At the same time, if someone who is HIV positive is willing to take an HIV positive organ or die, then I think that's what you're going to do. I mean, it's someone's dying. It's an option that I think they should be allowed to have. And then it opens up the pool of HIV negative organs for HIV negative individuals. So I do think it's a positive step forward, even though it's one that just as a scientist, I would be hesitant to allow.
Okay, Marty, back to you with the chat room questions.
Let's see. That's one where I can't pronounce half the words.
I know.
That's the one we want to hear then.
You just sound them out. I can figure it out. I had to close the chat thing because I was getting audio from there and here, and I was getting audio later. Just sound it out.
It's so hard to tell. Can't use immunofluorescence or something like that to identify reverse transcriptase inside of the dendritic cell to confirm HIV infection?
I know. It's not my thing. It's not my field. All I know is that there are lots of ways that you can test whether a cell is infected or not. And actually, I did a
So
little
essentially, the person was asking, "How do you tell if something is infected?" Or
yeah, there are
because
lots
I
of ways
understood
to tell.
absolutely nothing.
You can infect some cells and then wash them really, really good, and then see whether they start producing more virus. And they might produce some. It might be like there are lots of ways that they have gone through, whether these cells are infected or not, and it's still a point of contention. That being said, it's like I've actually infected some uninfectable cells before. I was using an epithelial cell line. So these are supposed to be skin cells. They were like skin cells. And that's not a target for HIV. But what I did was I wanted to make sure before I used them in an experiment that they really could not be infected, because that would have messed up my results. And so I put a ton of virus on these cells, and I used a kind of virus that I genetically modified so that the cells would glow green if they were actually infected. And I got some green cells, but I didn't verify, whether these were actually infected or not, because maybe it was just sort of like a random event. Like, it gets complicated. Things sound easy on the surface, but once you really get in there and want to scientifically establish something as a fact, the threshold of evidence is pretty high, and you have to convince a lot of people that don't necessarily believe you.
And in that case, the cells just misbehave.
Yeah, sometimes they do. And especially when, like, these dendritic cells, these are primary cells is what we call. You have to get them directly from a patient. And so there's a lot of characterization you have to do of those cells, and primary cells are just kind of weird to deal with anyway. So it's a battle. I can try to find you somebody in the field if you guys are really interested in how to address a point of contention in science.
All right, Marty, just keep with the chat room questions, because there's so many tonight.
Okay, let's see if I can find one. Another idea for a cure that I assume someone has thought of and concluded doesn't work. Isn't that exactly what I asked before, by the way? Could HIV potentially be cured using some type of dialysis?
Yeah.
Really, if you want to know my idea on the best chance of a cure at this point, I give a lot of crap to some of these HIV researchers who they go fishing in patients for these broadly neutralizing antibodies. So in particular individuals, they will make an antibody that will neutralize like 90% of the HIV variants that it's tested against. I get annoyed with these people because it hasn't really informed the way we design HIV vaccines. And ultimately, these people that make this super awesome antibody ultimately die of AIDS anyway. It didn't help them at all, because in that patient, it's just another selective pressure. It's not thinking that, I can neutralize all these viruses that I might be. That's not the way it works. So these scientists have found a whole bunch of these antibodies that are really good at stopping HIV. The problem is we can't make someone make that antibody. What you were showing before that little talk I did on antibodies, the randomness of how your immune system responds to a vaccine. I can't force you to make a particular antibody through a vaccine. What I can do, and what some researchers are doing, is they're genetically modifying viruses to deliver the genes that code for these broadly neutralizing antibodies. And so it's a kind of gene therapy to where I can't make you make this antibody naturally, but I can force some of your cells to make it by giving them it's cheating. It's giving your immune cells the right answer. They don't have to study. They don't have to work hard in school. They get the right answer. You're giving them the right answer. And they're working on that now. And I think that, 15 years ago, I would have said you were nuts, that gene therapy was a viable option to stop HIV. But at this point, gene therapy is becoming more widespread, and we've got something cool to give these viruses. I think it's a good idea.
Okay.
Cheating.
Could HIV and other viruses combine to make a super virus?
No. It's like saying, can like a giraffe and a blue whale recombine to make a super long neck?
Kind of sounds like
whale.
it, but I think that this is something that people like myself who don't know a damn thing about this kind of think about, like, for example, could HIV suddenly evolve into some airborne form or something? I mean, yeah.
It just for perspective, it's like saying, you know, could HIV evolve wings? You know, if evolution's so awesome, why don't humans have wings? We get that all the time. It's kind of similar. Like, evolution doesn't really work that way. Though that being said, it's not as if, especially retroviruses, they can't steal. So retroviruses, I'm trying to think of a way to describe this, are functionally diploid. So there's two copies of the genome. And so when it's getting turned into DNA, it hops back and forth in between the two strands. And that's neat in and of itself. We get gene duplication and divergence, and that's where a lot of mutations occur and how the quasi-species builds. It's not just the mistakes that are made. But theoretically, if there were something around, perhaps it could acquire genes from other RNA that was. It's not to say it could never, ever happen. And actually, we see in our genome some, not just humans, but like bats, I think it was, an endogenous virus that was non-retroviral. So it was just a virus that was around when reverse transcriptase, the enzyme that turns RNA to DNA, was around, and it got into our genome that way. So crazy, freaky things happen, but it's not something that I would be immediately concerned about.
Okay.
All right, just keep flying through them, Marty. We've got plenty.
I have one that I wanted to jump in with. Not to interrupt you, Marty, but it's a feminist-related question, so I thought I'd be the one to ask it. Matt's asking, he said, I'd be curious to know what it's like in the field for women. If I'm not mistaken, Abbie has gotten into some spats with feminists in the past, and I'm much more impressed by her feminism. In other words, just being a strong, intelligent... Oops, it moves. Sorry. In other words, just being a strong, intelligent woman. That said, are there lots of other women doing this type of research? Do you feel that there's any discrimination, no matter how light it might be?
My previous laboratory, the majority of the members were women. My current laboratory, the vast, we have one guy in my lab. It's literally all women. And not just we're not regular women. We're really loud, opinionated women. Right. So that poor guy, we feel so bad for him. But in my personal experience, no, I can't think of one incident where someone has said, you know, their little girl, run along and play with your dollies while us men do science. That's not how it works, and I don't know whether it's because of my particular field. This is HIV research. We don't have time for those games. People are dying right now. I don't care whether the idea comes from a man or a woman or a donkey. I mean, we got to get on this. And the thing is, too, it's like the market is as such. You know, if you're going to not have someone in your lab because they're a female, even though they are incredibly talented, they can go to another lab and be incredibly talented, and it's their loss, not the person who lost the job. We need great people in this field right now. So personally, no. Yeah.
Do you think that's fairly common across the board, not necessarily just in HIV research, but is it probably the same throughout?
You know, I can't speak for the other fields. For all I know, in certain branches of biochemistry or something, it's terrible, or astronomy, it's terrible. I can't speak for those, and I can't even really speak for other people in HIV. we actually talk about this sometimes in the lab. The people in the lab my age and younger, we don't see it anymore. And the older women are, thank goodness, because we had to deal with shit. And it's a relief that, what they went through, the younger people these days don't have to put up with that shit anymore. And it's a luxury. I love it. I think basically we're judged based on our science, and there's so many reasons to attack someone's science that you don't really need to worry about their skin color, gender. And, even especially with HIV, some of the biggest names in HIV research are women. And man, some of them, if you go to them trying to get support just because you're a woman, I don't think they would necessarily respond well to that, because they are big names, big personalities, and it's not centered around their gender, it's centered around their science. I wouldn't go crying to them about anything. They're the ones I'd be most scared of, actually.
Excellent.
Hey, Marty.
Okay. Could stem cells play an important role in curing HIV?
I don't know. Obviously they kind of do in the bone marrow transplants. But it's not something where we would just want to make more CD4 positive T cells from stem cells, because then you'd just be making more HIV targets. And, maybe if we could do some of that gene therapy I was talking about and kind of pre-program those cells before they went in. But, I don't know. I haven't been following that. There might be some people out there doing some cool stuff, and I just don't know. We will have to PubMed that one.
Okay.
What about swine virus? It's a combination, no?
That's influenza. I totally know what you're talking about there. So
I don't. I know about swine flu. Is that what he's talking about?
Possibly. So very, very simplistically, there's certain kinds of sugar on human cells, and there's certain kinds of sugars on the surface of birds, and some kinds of flu, like bird sugar, and some kinds of flu, like people sugar, and pigs have both kinds of sugar. And so what happens is a pig could be infected with a bird virus, and it changes a bit, and then all of a sudden it can infect people with our sugars. That's a very simplistic way of putting it. And so, so a swine flu is just a kind of influenza that has bits and pieces of its genome that look more like a bird virus or more like a pig virus, and not so much as a people virus, but it's infecting people. And the trouble is sometimes some of the bits and pieces it takes, the humans don't necessarily, it's not like, I got the flu when I was 4, so when I'm exposed to flu later, I kind of have a little bit better immunity to it. It's a kind of flu that we've never been exposed to before, so you're going to get sick from it. And that was the trouble initially, I think, when that crossed over, is nobody had any kind of immunity to it before, so if you got exposed, you got sick.
Okay.
Is that all the chat room questions, Marty?
I'm sure I've skipped several.
Okay.
Because there are so many of them.
Yeah.
Actually, I just have one quick question just on endogenous retroviruses. Have any of them have been found to have function in the human genome, either a good function or a harmful function?
Yes, yes to both of those. So one of them, that protein on the outside of viruses I was talking about that it uses to infect our cells, it's called envelope. It's a target of the antibodies. We actually stole that gene from an endogenous retrovirus to make placentas. So what happens, let's say an HIV-infected cell, it will have this protein on the surface because the viruses are going to bud off and incorporate that protein. So this cell has a bunch of that protein on the surface. If you put it next to another target cell, those proteins see the target cell and will start fusing. They don't know that they aren't in a virus. They just do what they do. And so what you get are two cells that smush together to make one big cell with two nuclei. And then they'll smush together with another one and another one and another one to make these big masses, these multinucleated blobs called syncytia. And our genomes capitalized on this to form placenta, like the connection between mama and baby is this big syncytial blob that we totally stole from viruses. So that's one case where it was capitalized for something good, and it happened in all mammals. That's the vast minority. That's the one out of the thousands and thousands and thousands of endogenous retroviral proteins and promoters in the human genome. Usually they're quiet, and that's awesome, because when they're activated on accident, that's when things start going bad. It's usually a cancer. I think it's either Hodgkin's or non-Hodgkin's lymphoma has been directly associated with activation of an endogenous retroviral promoter. And so giving the cancer patients drugs that kind of shut up that promoter has a beneficial effect on cancer treatment. And then, man, everything from schizophrenia to multiple sclerosis, all of these have been associated with wayward activation of an endogenous retrovirus. They're junk, and when they're quiet, it's good. When they start making noises, it's bad. But once they did something good.
Okay, we're coming up to the last few minutes, so it's time to wrap up. So, as always, we let our guests have a little soapbox moment. So, Dr. Abbie, if there's any you want to promote, anything, message you want to send out to the audience listening in the chat room and on the podcast.
No, I'm just going to repeat what I said before. Stay in school, drink your milk, wear condoms. Unless you're lactose intolerant.
I'll just go around the panel. Kitch, your final thoughts there.
Yeah, that was a really great. Thank you to Dr. Smith for coming on the show and giving her wealth of experience and study to us. So it's been brilliant. And and wear condoms.
All right, Sheila.
It was fantastic to get a chance to chat with you today. Absolutely fascinated with everything you had to say. Can I go ahead and do my plug?
Yep, you can do your plugs. Yep.
Excellent. I want everybody to kind of check out Facebook. We have the 3rd annual protest against hereditary religion tomorrow. Lundy's going to join me for a discussion about genital mutilation, among other things. So make sure you tune in for that. Also, when I'm not here, I'm also on A News. So you can find me there. Follow me at Sheila Blackadder on Twitter.
Right, I missed you. I missed you when I was on A News. You were sick or something. You weren't on. Aw.
Yeah.
Okay, Marty.
Yeah, I understood absolutely nothing of this. I suck at biology. I clearly need to brush up on that. Maybe I should actually read through a high school biology textbook at some point.
Read my blog. I try to take baby steps.
Yeah, it's just that I want a more complete thing. I can't stand pop science versions of it because I usually want a more complete understanding of a field when I look at it. I'd rather have the bare basics than a few details of really, really awesome stuff, you know. But we'll see if I ever get around to that.
Okay, and Wolfie.
Oh, I want to thank everybody for this, and especially Dr. Abigail Smith. And apparently Marty was not the only one who was having trouble following this. My knowledge in biology is probably just better than Marty because my last couple of years in college did include bio labs and the fundamentals of biology and all that stuff. So I was following along quite well. But there are a few things that I need to get up to speed with, so I will check out your blog. And
I've got a contact page too. You got my email readily available. I kind of suck at responding to emails, but I try.
No worries, no worries. Anyway, and I guess I will close up this by saying, great podcast. I look forward to the next show, and especially the podcast that Sheila just brought up. And it's all up with staying in school, drinking milk, and condoms.
That's the main message to take away tonight. Everyone, just in case, all the links are down below to subscribe to our podcast and the YouTube, the Facebook group, if you want to keep up to date with us. We're back next week. The scheduled guest is the Humanist Association of Northern Ireland. We've got some of them arranged to come in. So hopefully that should be very interesting given, you know, the history of Northern Ireland and that for obvious reasons. And just to let everyone know, our other podcast, the Antimatter Bandits, we're recording tomorrow and we're doing a commentary on the Phantom Menace. So that will be up and the website will be up soon.
It's going to hurt.
Yeah, it is.
Why? Why would you do that?
I don't know. It's weird with this podcast we do, Abby. We've tried to do commentaries on good movies, but it's not as entertaining as when we start ranting and getting angry with bad movies.
That and we're just a bunch of masochists.
Yes, we are.
Yeah.
All right, everyone.
Apparently.
Thanks for tuning in, and we'll see everyone next week. Hope you enjoyed the show, and like I said, subscribe and follow us down below. Thanks very much.
Bye.
Bye-bye, everybody. Thanks for tuning in.
Yeah.
Because other individuals' chances of acquiring HIV are reduced when everyone is on the drugs. So really right now, the goal is to get as many people on the drugs as soon as possible to help control the epidemic.
Right. Is there like an issue with the patients who are on the drugs? Does the virus kind of adapt to that, to where the drugs become as not quite as effective or eventually ineffective against it?
Sometimes. So you know how I was saying that HIV behaves a little different than the other viruses we've dealt with? Well, what happens is every time HIV replicates, each one of its little baby viruses is a little bit different than the parent strain. And so when that goes on over and over and over, what you get is a big cloud of viruses where they're all related to one another, but each individual one is distinct. They're all individuals. So what happens is you will give someone an anti-HIV drug, let's say AZT, and that will wipe out most of the cloud of genetic variants. But it's not going to get all of them. Just by chance alone, there are going to be variants in that cloud that are resistant to AZT. And so what we found out very soon after AZT was developed was that you can't give someone just one drug at a time, because then all that happens is you wipe out all of the viruses that are unable to deal with the drug. You're left with a population that's resistant, and then the population expands to form a resistant cloud of viruses. And so that's the idea behind having people on 2, 3 more drugs at the same time, is that it reduces the chances that there's going to be a variant in there that can resist all of the drugs you're throwing at it at the same time. But ultimately, yeah, how I got into HIV research.
Good story.
Yeah, no doubt. Where are you hoping that this field will take you? I mean, what is is there like a specific, are you working on are you wanting to be part of like the team that finds the cure for HIV? Or, I mean, what is your goal as far as work,
really, as a scientist, I just love studying HIV as a virus. I think it's fascinating. It has evolved millions and millions, if not billions of years to be absolutely exquisite to do what it does. And just the way HIV does things is so different than the way, for instance, giraffes or oak trees do things. Just studying the virus itself, I think, is incredibly fascinating. At the same time, it's incredibly destructive. You don't read about oak trees killing 27 million people a year. Really, that is what all of my research focuses around, is studying the virus and hoping that when we understand the virus better, we can figure out how to make an effective anti-HIV vaccine. Because right now we don't have it. I've said this before on other podcasts. Like when HIV was first discovered, scientists were like, give us a few months, but this is a virus. We know how to make vaccines. We're on top of this. Just give us a minute. And here we are 30 years later without an HIV vaccine. And that is because HIV behaves in a manner different than the other viruses that we do have vaccines for. So just the way it replicates, the way it evades our immune system is different than, for instance, polio or chickenpox or other.
And good evening, everyone. Welcome to Trolling with Logic. It's the 18th of January 2014. I'm still getting used to 2014. And it's 7 p.m. here in the UK, 2 p.m. Eastern Time over in the States. Just to let you know, you can call in later on in the show at the Skype contact Trolling with Logic. And as a reminder, when you call in, you grant us permission to use your likeness, because the show is later uploaded to iTunes and YouTube for later viewing. So no funny stuff with the DMCAs. Also, the recording of this show is public domain, so feel free to copy, share and distribute it as you want. Just link back to us as a little courtesy. And I think that's all. That's the legal stuff. So quickly around the team, and welcoming back after another long layoff, Wolfie, how you doing?
I'm doing good. Oh, it's great to be back. Good to see everybody.
All right. And Marty, how you doing?
Yeah, I'm fine, but my webcam isn't. So I apologize for that.
And as always, Sheila, how's you?
Hey, glad to be back.
And last but certainly no least, Kitch, what's up?
I'm grand. How's everybody?
All right. Yep, we're doing great. So you did have an intro for our guest all written up there, so take it away.
Yes, I've just written up a brief intro. I just want to welcome for her first podcast as Dr. Abbie Smith. She has defended her thesis on novel strategies to understand HIV treatment. She runs her own science blog on Science Blogs on ERVs, and she has debated for her sins a few creationists such as Charles Jackson and Steve Kern. Yep, that's just the brief introduction I just written up there.
All right. So as always, I think Sheila, you can take it away with the introductories. And sorry, let Abbie say hello to everyone first. Sorry about that.
Oh, hi guys. I'm really glad you all invited me to talk. And like he said, I just got my PhD. So this is the very first podcast that I am Dr. Abigail Smith. So, yeah.
The doctor is in the house. So Sheila, back to you. So we'll let you start with your introductories.
Awesome. Well, let me start with congratulations on your doctorate. That's awesome.
Thank you.
Can you give us a little bit of background? How did you get into the field that you're in? What made you decide to go the direction you did?
That's interesting. So when I was an undergrad, I was like a hardcore pre-med student. I was planning on going to medical school. I came from a small farming town, so I thought if you were good at science, you either became a physician or you became an engineer. I didn't really know any PhDs in biology. I thought they did like forest work and stuff. That's what I thought a PhD in biology did. But when I got to college, I was around actual PhDs in biology who did different kinds of biomedical research. And I got an opportunity to do a summer research program in virology and HIV research. And I only did it because I thought that having, HIV research on your medical school application would look really, really good. And then it turns out I loved it. I had so much fun. And I had more fun. It was more entertaining to do that than the stuff that I was doing in hospitals shadowing physicians. And so I was like, maybe I shouldn't be a physician. Maybe I should explore this other opportunity. So I took a couple of years off to be a technician, a research technician in an HIV lab, to really see if, you know, if I really did love this. Did I want to do this 9 to 5 plus every single day? And I totally love it. I don't want to do anything else. This is dream job, so much fun. They're vaccines that we have made.
Right. I guess, can you explain a little bit the difference between, like, there's like a cocktail of drugs that people that are infected take. Can you kind of explain the difference between, is that more of a just kind of keep it under control sort of thing? What would be the difference between that and a vaccine?
Yeah. So the antiretroviral drugs are keeping the virus from replicating a lot, from making lots and lots and lots of baby viruses. But just the way HIV works, it's always going to be there. You can't get rid of every last virus, but the drugs control it. Whereas an anti-HIV vaccine, you would get before you were ever infected, and it would prevent the virus from ever getting a foothold, so it can't be eradicated. That being said, an idea that's been floating around is something called a therapeutic vaccine. So let's say you were already infected with HIV. We might still be able to give you an HIV vaccine that has a therapeutic effect. So if, for instance, we could figure out what the more aggressive variants of HIV looked like, even if you were infected with HIV, we could give you that vaccine, and then that would prevent the more aggressive ones from evolving. So we have the therapeutic drugs, a theoretical preventative HIV vaccine, and then a theoretical therapeutic HIV vaccine. Really, right now with the drugs, that's our best option. The anti-HIV drugs, when people take them, it's great at extending lifespans. People who are infected with HIV, their lifespans are approaching those of uninfected individuals because the drugs we have now just work really well. And actually the lifespans of individuals in a community where the people with HIV have access to the drugs, the overall lifespan is increasing